So good day. I'm really excited to present a little bit our work that we have done using the custom mini-genes of IDT to produce indeed peptides in another way in bacteria. So first, in fact, I want to address the question why we need peptides, and I think it's very clear that we want to interfere with discrete protein complexes, while you might want to maintain other cellular functions of your protein, which may essential. I will come to that later. In addition, it's very important that protein domains might fulfill certain biological activities in the full-length protein, and it's good to study those in dissection or isolation of other functions of the proteins. Furthermore, there are now many more examples that they might be good models for with therapeutic potential. And as such, they can be delivered into cells because of cell-penetrating sequences, or they could be also even targeted to certain compartments. I think also the fact that one now can use peptides to create mimetic molecules that retain the biological activity is very, very promising. In fact, I want to go a little bit deeper into the fact to interfere with discrete complexes while maintaining cellular functions. For instance, you want to discover novel or non-canonical functions of an essential protein. The knockdown of this protein might not be a good idea. It might require longer periods, hours, days, and in fact some cell death or other processes might arise before you can perform your desired analysis. So in fact, selective interference with a domain of the end-apoptotic protein might be much more convenient, and this typically would require much shorter periods, and probably in these cases cell death would not become a major issue. Now I want to give you a little bit background on why we needed some of these peptides and invested in new methods. In fact, our group is studying calcium signaling in cell death and survival, and in fact I always compare calcium as the Roman god Janus here in the middle, in the sense that it's a two-faced god, and dependent on the profile of the calcium signals, they might either promote survival or promote cell death. And in fact, it's very clear that the regulation of these signals is modulated by protein-protein interactions with the B-cell2, anti-apoptotic B-cell2 family members as very important proteins, both at the mitochondria, but more and more evidence also at the endoplasmic reticulum, where they target intracellular calcium release channels. And in fact, we, and with the use of peptides, found selective functions of two neighboring proteins of B-cell2 and B-cell-XL that have discrete functions on these calcium signals that be oscillatory or to inhibit the complete signal here. Now the IP3 receptor, if we think about this protein and you work with this, well, a major challenge is that it's a very big protein, 2,749 amino acids. It's a tetramer, 1.2 million Daltons, so which means it's really challenging to work with it. And you can nicely see here, for instance, the IP3 receptor and an electron microscopic view to see the higher order structure. And so in fact, there are discrete domains in these channels which can bind the activating molecule, which have discrete regulatory domains, but also at the C-terminal a channel pore. And in fact, this is not even an updated picture, but it's, or I want to say a complete picture, but we have really many proteins that target really distinct sites at this protein to modulate its function and its calcium flux properties. So you really see, if we would knock down this protein, we would affect many other of these functions. Okay. So what we studied is the anti-apoptotic protein B-cell2, and with the use of peptides, we could show its binding to a very discrete region, a 20 amino acid region that is located in the center of this protein. In fact, here is some data just to show you the challenging approaches that we needed to go through. Is that the IP3 receptor, it's of course when you want to study protein-protein interactions, you might want to chop it up in different pieces. And in fact, we did this in a way that we retained the molecular structure, because with tryptic digestion, we knew these domains were formed, and we used them and express them as a GST fusion protein. And here you see each domain here of the IP3 receptor, which is expressed and purified as a GST fusion protein. And in fact, using this protein, we could identify the BCL2 binding site. But okay, at this point, we are at 600 amino acids, which is still far from the identification of the site. So in fact, we went on and used smaller domains, and that's how we came up with a effective, very short peptide of about 20 amino acids. And the cool thing about this peptide is that the single peptide with 20 amino acids can completely block the binding of BCL2, which you see here, to the IP3 receptor in an immunoprecipitation. And if we add this peptide, in fact, this binding is completely gone, which is really, really very nice proof. In fact, we are now using this peptide also in cancer cells to promote pro-apoptotic calcium signals. In fact, another domain, we also mapped the domain at the BCL2, and in fact here in yellow, or you see here the crystal structure, and here in yellow you see the unique BH4 domain, and in fact, using similar approaches, we were able to show that the BH4 domain is really the domain that is targeting the IP3 receptor domain in this pull downs. And in fact, we could also tag this peptide and pull it down with a Fitch label, and then we could completely pull down the IP3 receptor. So the single peptide is able to pull down this IP3 receptor. So it's we really identified in a protein of about 2,700 amino acids. We identified a discrete locus, and we did the same for BCL2. And of course now this offers more tools to really tweak the binding and to interfere with the binding. So in fact, why did we then go on and looked for alternative methods? So of course, research-wise, we wanted to explore the BH4 domain's characteristics and functions, develop mutants and variants. We wanted to develop membrane-permeable variants for cellular applications, but also wanted to develop some affinity purifications and detections for immunofluorescence and Western-blotting. So for us, it was clear we needed to invest in an alternative method to develop peptides. So what do I mean with this? Is that we have high flexibility, that we can adapt it to our choice. It's fast. We would have an unlimited supply, and we could maybe make longer peptides. And in fact, that's how we came up with the use of the custom mini-genes from IDT for developing peptides in a biological system. And the reason for us was very clear. I mean, first of all, you get the feedback on the codon usage, and there is an important quality check before the whole procedure is initiated. And in fact, we obtained very high quality vector constructions which have been quality controlled at multiple levels at a very fast turnaround time. And in fact, this system now allows to easily create novel mutations or insert new tags. And in fact, it's possible to insert new tags to generate longer peptides at additional low cost. These additional costs are in fact relatively low, and certainly if you would compare to chemical synthesized peptides. So how do you start with such a project? And in fact, it's important to realize what you want to achieve. And in fact, there are different choices one needs to take into account, and it starts with the choice of your vector of expression and expression system. And I have to say, when I prepared this slide, I think most people will in fact stick to the bacterial expression, because yield is a very important issue, probably more important than the post-translational modifications and the folding properties, but might be important if you work with longer peptides. The folding, these modifications might be important, and you might switch to eukaryotic systems, and there are different possibilities there. On the other hand, you also need to think about the choice of a tag for purification, because of course if you do a biological expression and purification, you need something to purify, and affinity purification is really convenient. So in fact, what we recommend is that one would use a backbone vector in which you already have a tag present, which might be GST, maltose binding protein, but also GFP. There are pretty good GFP columns and GFP purification methods. But in fact, And in fact, these larger proteins might in fact also help with the stability of your peptide. On the other hand, I think it's also convenient to use shorter purification. The 6-his, the HA tag, the FLAG tag, MIC, V5 tag might all be convenient to tag your peptide. On the other hand, if you want to have the flexibility, you could also put it in your insert. But of course, there I think it's better only to have the shorter peptide, shorter tag present. These are, of course, tags for purifications. Of course, one can also use these tags for further detection and assessment. And so we also need to think about that. Do you want to maintain these tags during your detection procedure? In that case, you want to get rid of the longer tags, and that's definitely a choice of detection. But I think an HA flag tag is very convenient for further assessment. Also, if you want to use your peptides in cells, I think you need to think about a cell-penetrating sequence. I'm not providing a full list. There are people that are more expert than me on the cell-penetrating sequences, but we have had good results with the TAT, so which is the HIV from the HIV virus, the TAT protein of the HIV virus, and the TAT sequence. It's about 9 amino acids. It's polylysine, polyarginine-rich region. It's very convenient, and it's only 9 amino acids, very convenient to put in front of your peptide. Antennapedia is a little bit longer, but also very good. And I know there are now also modified versions of the TAT and other penetrating sequences that you could put in. Of course, if we think about tags that we want to remove, we need to insert cleavage, protease cleavage sites. And many are present, can be used. Thrombin, factor Xa, precision protease, TEV protease, all are excellent choices to work with. And it's dependent on you what you think is more easily achieved, and also you can easily remove. On the other hand, I also would maybe like to mention that when flexibility would be very important for you as the prime criterion, you might consider using the gateway system in the sense that you create an entry clone. I'm not going too much into detail, but you could create an entry clone, and in fact this entry clone, one of the steps, this could be done by IDT by having this vector synthesized with your digested DNA fragment containing your peptide of interest. And in fact, once you have the entry clone, you have the full scala of all the destinations vectors where you can recombine your protein with, and you can create really in a flexible way the different systems. So that's maybe one thing one might consider. Of course, when we talk about biological production, and I will mainly focus on the bacterial production because that's what, in fact, we had a lot of experience now, or some experience with. We chose to use the IPTG-inducible expression system, which you can find in the PGEX vectors or the PET vectors. We used a strain that's really convenient for that, and BL21D3, which is really excellent for doing protein and peptide expressions, and I think that's really good. There might be other strains, but we stick to that one. Then I wanted to say something about growth conditions. I mean, of course, everybody or most people are aware of the standard growing conditions in which you can dilute and then have the same day expression at a lower temperature with the IPTG. However, since we worked with difficult to express proteins, we needed to develop a new protocol. And in fact, we found, and this is convenient maybe also for peptides that might be less soluble, if you do a double overnight step, after the 37 degrees expression and the dilution to 22 degrees, then you can induce your expression at very low temperatures for 16 hours. And this really gives very stable proteins. We really found this is sometimes very, very convenient, and it doesn't stress the cells too much, and it really gives them the time to properly express the proteins and so on. Of course, the optimization of the IPTG concentrations is also important in this perspective. Then, for affinity purifications, one should check the solubility, whether it's in the supernatons or the pellet. And when your peptide is in the inclusion bodies or in the pellet, because it enters up into the inclusion bodies, that might not be too bad. Of course, it's convenient in the supernatons, but sometimes these pellets are really protected against protease degradation. And in fact, we have some good results by solubilizing these pellets with sarcosyl, and then these proteins still reside in a good conformation. You also should check the integrity of your fusion peptide, and in fact you should use the text that you can then check via Western-blotting. And I think you should check the level of your fusion peptide, and of course you can use colorimetric assays. Sometimes, okay, these might depend on the presence of certain amino acids, and of course with your peptides one should see that we do not get any caveats, but you could also load a golden standard on like an SDS page and use this golden standard and to compare. On the other hand, we also need to, after the purification, we need to elute, and I want to remark that the buffer that is used for the purification is often, and the elution is often not very good for further assessment, so we also recommend dialysis against the buffer of choice. One should also remove tags, and then we should confirm the quality control, which is concentration, integrity, and the determination of contaminants. And I want to make a special case about the contaminants, because in many cases you might see that your GST fusion protein or your other proteins might contain some bacterial chaperones. And in fact, I will recommend you 2 papers how to deal with these aspects. Then there are additional concentration and purification steps if needed, and maybe the removal of your protease. If you, for instance, use the thrombin, of course the thrombin might be still present and you need to remove it or at least inactivate it. I want to bring to your attention these 2 papers because we also face sometimes the problem of a DNA K and the GRO-L that is present in our GST fusion protein purifications of other protein purification, and in fact they have a nice strategy how to get rid of those. So that might be a thing to consider if you want to proceed with these procedures. Okay, so let's go to our example of the BH4 domain of BCL2. So that's the vector we introduced. We have a PGEX6P2, it has a precision protease site, and we cloned the TAT-HA-BH4. And I want to bring up the potential, which we did not do, is to include additional protease cleavage sites between the 2 tags, so giving you maximum flexibility. These protease cleavage sites could be one of these 4, which are giving you a cut in after some amino acids, leaving you the yellow part present in your peptide. Okay. We would recommend, if you use this vector, to clone the BAMH1 EcoR1. In fact, you have the complete choice of your codon, so there is no reason to use other restriction sites. And in fact, the reason for this is very clear: is that if you would use other sites, I mean, these residues will pop up in your peptide. Of course, if you're working with small sequences, you want to avoid the unwanted residues or that are present, and in fact in this case you still have the glycine proline leucine that would be present. Okay, and in fact I found this also maybe a disadvantage, this proline residue, because we know prolines might affect the structure, and in fact we might have interference with some of these residues in our assessment of the biological activity. Okay, it's a consideration one can make. We could introduce other protease sites that are convenient there, and it's definitely solvable. This is just the 3 mini-genes we ordered and that we used in frame, and in which we then have the different tags and the sequences present, including a stop codon in between these restriction enzyme sites, but just for your interest. Okay, so we were able to very nicely purify these peptides, and in fact I just show gel coat blue staining, in which we purified the GST TAT-HA-BH4 of BCL2, and in fact we could cleave the fusion protein. That's very pure. We could cleave it here, and we would obtain the pure GST and the TAT-HA-BH4, which is about 5 kilodalton. Okay, we also did a quality control, and which is the Western blot analysis. You might see here we get with the anti-GST, we can nicely see the protein, and in fact the 34 is one of the BH4 domain proteins, and you see also the shift with the parental GST, which is this one. So it indicates that here we have clearly a good expression, and in fact we could confirm this with an HA tag, and I think the HA tag is really nice because, okay, it's the one tag we inserted into a protein, and in fact it's present here, but not in the parental GST. Okay. On the other hand, what we noticed when we compared the supernatant with the pellets and we solubilized with sarcosyl, we found in fact much of our protein present in this sarcosyl pellet, and I think the reason for this might be that we have very prominent expression, and also some of the BH4 domain might be hydrophobic properties. So what was our optimized workflow, which gives you some nice view? So we did the same for BCL2, BCLXL. We could purify the GST TAT HA BH4. We could cut it, giving us GST TAT and TAT HA BH4. And then, of course, we needed to get rid of these other proteins, the GST, some of the contaminants, and in fact we did two additional steps, centricon purification and another purification on the GST column, and in fact we could obtain relatively pure TAT HA BH4. In fact, here I estimated it at 70%, and in fact we obtained 0.2 to 0.4 microgram per microliter, which of course might not be very high, certainly if you compare to GST, but it's about 10%, 20% of this yield. But we think this is partly due because we know that the pH4 domain, it's not very soluble in a water-like solution. So when we ordered it, we needed to dissolve it very well in DMSO, and it was not well dissolvable in water. So I think there might be some issue when you isolate the peptide that it's not fully soluble, and therefore we lose some during this process. In fact, we could then confirm its integrity by doing an anti-HA Western-blotting, where we here could purify it, and we could test it on Western-blotting. And here in the first experiment, we did some pull downs, which were not successful with the IP3R domains at this case. But okay, with this experiment, I just want to show we can nicely see the peptide, and it's very clear that we can use that. In fact, after some optimization, we were able to purify and to use this peptide. Here you have the input, which is a little bit overloaded, unfortunately, but here you can see that the peptide is binding to our IP3R domain in a relatively more condition, so it's a biological evaluation compared to the GST control or the beads control. And in fact, here we had some remnant of the uncut GST TAT HA BH4, which also popped up in these pull downs, which is in fact sometimes beneficial because the GST might help to stabilize some of the conformation because it's a big soluble protein. In fact, it might sometimes help to get the peptide more soluble. So it's not always a bad thing to have some GST there. Unfortunately, we, of course, you notice that we use the TET, and in fact this TET we used to promote cell entry. And unfortunately, that does not work. Okay, so the cellular uptake of the TAT HA BH4 we have here stained with an anti-HA antibody and then with a secondary antibody labeled with Fitch. And the red that you see is some of the phalloidine Texas red, which stains some of the actin fibers. So it really gives you a very nice view. The red is the surrounding of the cell, and then the green is the peptide, but in fact we didn't really see a difference between the no TAT peptide control treated cells and the TAT HA BH4. And therefore I wanted to raise the concern when one uses this precision protease, there is this proline that always will be in front of your peptide of interest. Okay, and this might really obscure a little bit the TAT cell-penetrating sequence. So I think this might be an issue that one needs to solve if one wants to use this method to develop bona fide cell-penetrating peptides. But it might also be an issue just because of this combination, and that it's nothing to do with the pooling, but we did not further assess that. Okay, so I wanted to maybe give you an overview of what are now the advantages, disadvantages of biological production versus chemical. So it's clear the workload for a chemical synthesis is nihil. You just have to order. But if you want to produce, it's high because you need to do some preparation, quality controls, activity checking. But there is the option of FPLC automation. So for instance, in our lab, we have an FPLC and you can run it overnight. And in fact, it's quite, once it's set up, it's easy. The cost, I think, for biological production it might be on the low side. For the chemical synthesis, this will obviously depend on the length. the modification, the yield, and the purity. I will have some comments about that. The yield, of course, and the purity are flexible choices with the chemical synthesis. When you want to end up with high amounts of very pure peptide, you will pay a lot. So here you could just tweak the purification until you get the desired purity. Also, some of your biological experiments might not need a high purity. If you have 70% purity, I think might be enough to assess some binding activities. Chemical properties of the peptides. I think with chemical synthesis you have the freedom. We really have hydrophilic, hydrophobic sequences, and they all sensitized very well, and we could dissolve them. For the biological production, I think that might be an issue, the hydrophobic one. So I think it's when one starts with this, I would stick to the hydrophilic sequences. What I really found an advantage of the biological production is your high flexibility and your high adaptability. What I mean with this, you could choose in the expression system, you could choose to remove text, but also the adaptability, you could choose to create novel mutations and so on. Because remember, if you go for a chemical synthesis, each peptide, each mutant is not a mutant, it's a new synthesis, and you will pay for this new synthesis. I also think the advantage is the supply. You have an unlimited supply of your peptide because you have the vector. You can make more of the vector, you can make more of your fusion protein, and so on. Whereas with the chemical synthesis, you really need to decide this in advance with fixed amounts. And this might be a problem because of stability. Okay, some prices, typically they go down when you order huge amounts, but we also noticed already that some of the stability of these peptides might go down even at -80. In fact, we now recommend to use DMSO to prevent some hydrolysis to dissolve your peptides. Here you could just remake the peptide if you think the biological activity is going down. On the other hand, I have to say that the risks in this case are much higher. Okay, here you could order a small amount, which is cheap. You could order some low purity and check some initial things, and okay, so there is a relatively low risk. Here with the biological production, you have to take into account the risk that is there, obviously. So I also want to make a comparison. If now I would need to choose some of these peptides, what I would do, because okay, I want to give a balanced view. I think for shorter peptides, hydrophobic sequences, I would say, no question, I would go for the chemical synthesis. Shorter peptides, they are very cheap. Hydrophobic sequences are hard to sensitize. So I think there it's a clear advantage for the chemical synthesis. With the longer peptides, okay, it's balance. Okay, you have to think, do I want to spend the effort there? I think it's definitely worth trying the biological production, because take into account that long peptides at high amounts and purity become very expensive. I have to say we have ordered them. It's also convenient, so you have the choice. But for some things, I would say the investment in a biological production might be very well worth it. If you want very high purity, for instance, you would go for NMR and so on, I also would not take the risk. I would go for the chemical synthesis and order the high purity peptide, because you're 100% sure what you order is what you get there. Whereas, okay, with the biological production, okay, there might be problem with contaminants. Okay, so I would then go to the chemical synthesis. However, if you think that flexibility and adaptability are prime criteria for you, I would give it the advantage to go to the biological production, because you have the removable tags. You could use the tags in different assays. Immunofluorescence we have shown, although not successful, but okay, it's definitely an option. Pull downs, immunoprecipitation, some other assays are very, very good. Also mutations, recloning, it's the adaptability. You have the freedom to use. Okay, I think the biological production has a clear advantage. Take into account that each mutation is a new order, it's a new chemical synthesis, so for the people there, they need to charge you again the full amount of a peptide, whether or not it's a derivative of another peptide. Okay, it's independent of that. And of course the supply. Okay, of course with ordering chemical synthesis, you could order as much as you want. Typically prices go down when ordering large amounts, but then stability might become an issue. Okay, and then even your cheap peptide might have degraded in the freezer while you treated it very well. Okay, whereas with the biological production, for sure, you have an unlimited supply that's very convenient, and it's definitely an advantage. Okay, I see I'm running I've finished with the seminar, so I definitely need to thank the people of the group. I have senior people, senior advisory staff like Humbert de Smet, Ludwig Michian, and Jan Parijs. And then I have really a motivated group of PhD students and postdocs, and especially I need to thank Giovanni Monaco. He really spent a lot of time purifying different peptides, purifying, checking their expression and so on, and eventually, okay, we could not use it, so I'm happy at least these tools now might serve for this purpose. So I really am very grateful to him. I also should not forget the dean of our faculty, which in fact also is a member of our research group. And finally, I need to thank the people of IDT really for giving me the opportunity with the nice article in their newspaper, Ellen, Sean, and Johan, and then the people of Byte Science Bio, which really helped me a lot with the preparation of this seminar, Gemma and Nick. So I really thank everybody. I'm open for questions.
Okay, thanks, Ger. That was a great talk. Thanks for sharing your work. I find it very informative. I just want to say that the picture of your lab, your group, they all look far too cheerful to be doing research, I think.
And freezing cold, it was in the winter time.
You all look too happy. Okay, so we now have a few questions for you from an audience. They cover quite a broad range of topics that you touched on. So if I just start randomly. At the end, you compared the biological and chemical methods. Did you try chemical synthesis of your peptides, either yourself or outsourced?
The chemical synthesis we always outsource. So there, in fact, we work together with Thermo Fisher and Lifetime. I have to say that we did not actually order the TAT-HA BH4 because I have to say, if you think about this, let's see here. Okay, this is a peptide I think of about 40 amino acids. That would really cost really a lot of money to just test this.
Yeah.
So in fact we chose this biological option because we would have the flexibility. So unfortunately we did not done it side by side. However, we have tested the TAT-BH4 BCL2 which was active. So but there I want to raise the concern that one needs to take into account that we have this glycine, proline, leucine, and I think also some of the glycine serine, so okay, that might be the difference.
Okay. When would you recommend using this method for producing peptides over the chemical synthesis? So you've maybe touched on it there with the length. Sorry, go ahead.
Yeah, so for sure, I think if I would go for the shorter and hydrophobic peptides, the chemical synthesis would always have my favorite. I have to say, when we wanted to use it into pull downs or other flexible methods, or we are producing longer peptides, I mean it's really expensive to go to the chemical synthesis. I mean it's really we have done it for one paper. I have to say, it's really like you order one peptide and that's it. So often we want to create mutants, we want to create other tags, so that's really not affordable for us.
Yeah, actually that links nicely to the next question. You've mentioned that this is good for producing peptides. Do you have any feel for the maximum peptide length you could make?
Actually, I would say that you could go up to 100 amino acids and even longer. Of course, then it becomes a fusion protein and not really a peptide anymore. So in fact, using this method, I think you have no restrictions. If you use the specialized expression protocol, I think there is no limitations. I mean, we go up to 300 amino acids longer, even 600 amino acids, but of course then it's not a peptide anymore, but it's your freedom.
Yeah, I suppose then you're looking at protein. Somebody's just mentioned I have noticed that you have not mentioned about sumo tags that do not leave additional residues. What do you think of them?
Yeah, that's a good, yes, that's a good point. Yeah, we did not, yeah, that's a consideration, yeah.
Okay. Somebody's asking, what about post-translational modifications in bacteria?
Yeah. that might be an issue, or it might rather be an issue that your post-translational modification is not present. Let's say if your peptide is only active when you have a modification, likely it will not occur in bacteria. For instance, phosphorylation and so on. It would be very strange if this would actually happen at this place.
Yeah.
So what we do then recommend is that you would either make a phosphomimic. If you really want to do the phosphorylation, you could make the serine into an aspartate or glutamate, or the phosphodet, or as I explained, you might consider using insect cells or even yeast cells. I know people who worked with yeast cells, and you might get good amounts still and get the post-translational modifications and fold it. Okay, it's always an isolated domain. Okay, how much does it consider relate to the real-time, the full-length folding? Okay, one never knows, but one could consider that. And I want to raise if one goes to the gateway system, I think it's perfectly doable. Yeah.
Sorry, what's the gateway system?
Okay, sorry. If one uses the system of I think it's solely in vitrogen, but it's with the L1, L2 entry clone generation that can be done by the IDT. Then you have the full scala of destination vectors in which you can target your gene. So you only make this once, and then you just recombine it to other vectors that might be suitable for you. So that might be an option.
Okay, so there is another option then.
Yeah.
Okay, so actually mentioning something on your slides, not necessarily related to the gene synthesis, you've mentioned sarcosyl a number of times for solubilization. I have not heard of this. What is it?
Okay. In fact, it's a mild detergent. It's a very mild detergent that is really recommended when you use GST fusion proteins. And in fact, the detergent solubilizes your peptide if it would be present in the inclusion bodies. Let's see. Okay, the sarcosyl. So it's present as a detergent, and you could solubilize your pellet and the inclusion bodies. And it's believed that it retains the biological activity and the structural properties of your peptide of protein or protein intact.
Okay.
So basically, it's a detergent, non-denaturing detergent. Yeah.
Yeah, I must admit I've heard of things like tween, but not sarcosyl either. So.
Yeah, yes, it's typical. It's a very specialized detergent, but it's freely commercially available, and it's really recommended when you do fusion protein purifications. Yeah.
Yeah, someone's just actually texted in. Is it N-laralsalcozine? Is that what you're? Is that
yes,
what it is?
Yeah.
Okay.
Yeah.
Okay, that's to know. I'd not heard of that one. Someone's asked quite an interesting question. This is a test for you as a professor of biology then. Does every bacteria generate peptides? And if so, is there a signature corresponding to each genus? That's quite interesting.
Well, of course, I have to mention that here we selected a specific strain. So when you're working with bacteria, what we are doing here, we are using the bacteria as a cellular bag, if I might say it in a disrespectful way.
Yeah.
In the sense that, in a crude way. So, which means that this does not reflect really the cellular biology of the E. coli. There might be, of course, when you are studying discrete species, distinct sequences that are produced by the E. coli, but here, of course, we use the modified E. coli strain, which is really suitable for these expression systems.
Yeah.
And so, in principle, it would rather depend on the chemical biophysical properties, biochemical properties of the peptide sequence, whether it would be able to produce the peptide and whether it would be soluble or not, rather than on the bacteria itself.
Yeah, because you get lots of different E. coli strains, don't you, for producing things like toxic proteins and
yes,
there's different types.
Yes, so here we stick to the BL21D3, but if you think about other strains that might be suitable for you or that have other proteases that they lack and so on, it's perfectly fine. As long as you can produce your peptide in it or your system, it's fine.
Okay, I'm going to read this question. TAT cleavage site peptide without any tags are expressed. Would the TAT sequence help the expressed peptide to penetrate the bacterial cell wall so that we can purify the peptide from the media itself? Did you get all of that? That's quite a long question.
Then probably I would go with secretory sequences, with secretory signals that you put there. I think it's a good thing. On the other hand, I would think it would, if it's secreted by the bacteria, you grow them in quite large volumes, so it would require quite some concentration and so on. But I'm not saying it's not possible. It's in fact a good idea. One might try.
Yeah, I mean, forgive my ignorance, but I've not worked with yeast, but I know people used to work with yeast because that exports. You can export your express protein in that, can you? Or is that? Oh, okay. I remember well.
There are possibilities in mammalian cells and so on to indeed have secreted sequences, and then of course that might be an option.
Yeah. Okay, so coming back to the gene synthesis, we've had a question. How difficult was it to obtain the mutants, the single mutants?
About the gene synthesis.
Yeah, so you had your 3 different sequences. Was it easy just to get the 3 different sequences?
Well, I have to say that so we just ordered the 3 different sequences. So if you just, like in this case, we wanted to create 7 mutants. I mean, that would be a huge effort to check all this. So in fact, what we did, we just ordered the 3 different sequences from IDT, so which was, just very convenient. I would say if you want to have single point mutations, you could just use this vector and then make in the mutations via the quick change methods, and that's very convenient. Only when you start to mutate, in this case, we mutated 7 amino acids, and the reason for this was because we identified 7 amino acids that were in a biochemical approach, in a modeling approach where the surface is accessible, so we mutated all of them. But in typical, you could simply then mutate one of those, and then it's convenient to do the quick change. If you go for 7, I think it's better to order it from the company, I would say.
Yeah, I imagine doing 7 mutations by quick change would take a very long time. Okay, so
yes, so it all depends on.
Sorry. Hello, Gert.
Hello.
Hello. Go on. Sorry.
Yes, I'm still there.
Okay, good.
Yeah.
Okay. We'll just have time for a couple more questions. What do you do if your peptide sequence is very hydrophobic, and what do you do if your 6 histidine tags, and what do you use? Sorry. And what do you do if you use his6 tag for the purification? Is there any possibility that the tag can be hidden? Shall I read that again?
No. Okay. I have to say that with the 6 his, we have had some issues, not particularly with these peptides, I have to say though, but I mean, I know this might be an issue that the his is not always convenient to work with and that it might be hidden. I would say with a short peptide, you would not think that there is such kind of problem. On the other hand, you might have the option, in fact, if I may, you could design the pet vector, you have the his tag, and you could insert some linker sequences between your peptide of interest and your his tag. And between the linker sequence and your peptide of interest, you could introduce the protease side. So this means that you have the his tag for your purification, and on the other hand, after the purification, you might want to remove the his tag and the linker sequence just by using the protease. I think that would be an option in that case.
That would be. So I actually have another question that may test your molecular biology as well. I promise we'll come back to the mini-genes. I have purified cleavable his-tagged hydrophobic peptide. I have tried to cleave the his-tag using cyanogen bromide technique, but the cleavage has failed. Do you have any suggestions to cleave the his-tag? Good one.
Yeah, Yeah, it's hard on.
It's a question out of left field.
No, actually, I have no. At that point, I cannot. It's when it's a hydrophobic peptide, I have to say we have experience. I think it might be issues they become an inclusion body. So I would say if you should check it's not aggregated. If it's not and that is the problem, that it cannot be easily accessed by or protected in that kind of thing. And in that case, you might want to use the Sarkozyl to get it in a more accessible state. That would be my recommendation, but I'm not sure whether this is a good answer. I mean, it's a trial and error at that stage, but I would say definitely with hydrophobic sequences, they tend to clog together.
Yeah.
So I would recommend to first optimize the conditions where they don't clog together, you don't have the aggregates, maybe use a little bit of the detergent. I'm not sure, even, yeah, that might be an option.
No, that's a good answer. That's a very, if it's all aggregated, then you can't necessarily access the cleavage site. So
but of course, the question might come from somebody that already he tried to, that it's not an aggregate and still it does not work.
Yeah.
So, yeah, I don't know. Yeah.
I suppose that's an in-depth, that could take a long time to answer that question.